If you want to know whether your body has what it takes to fight cancer, forget scrolling through your family medical records. Forget staring at blood work panels. Instead, pay attention to what is quietly living inside your gut at this very moment. A groundbreaking study published in one of the most respected scientific journals in the world revealed something that stunned the oncology community. When patients with advanced cancer were given the exact same immunotherapy drugs at the exact same doses, some went into full remission, and others didn't respond at all. The difference? It wasn't their age. It wasn't their genetics. It wasn't even the type of cancer they had. It came down to one specific species of bacterium living in their intestines. In this video, I'm going to walk you through exactly which gut bacteria have been scientifically linked to cancer immunity, why most people have dangerously low levels of them, and precisely what you can eat, with specific amounts, to help grow them back. Disclaimer: this video is for educational and informational purposes only. It is not medical advice. Always consult your physician before making changes to your diet or health routine, especially if you are undergoing cancer treatment. Most people think of gut bacteria as nothing more than digestive helpers, the things responsible for bloating or an upset stomach. But decades of cancer research have uncovered something far more significant. Your gut microbiome may be one of the most powerful factors determining whether your immune system can locate and eliminate cancer cells before they take hold. Your gut is not just a digestive organ. It is your immune system's command center, and the bacteria living inside it are either strengthening your defenses or quietly tearing them apart. I'm Dr. Leana. I'm an emergency medicine physician and cancer researcher. After years of studying the relationship between the immune system and cancer, I witnessed something that fundamentally changed how I talk to my patients. We've spent so long focused on treating cancer after it appears that we've largely overlooked the internal environment that allows it to develop in the first place. That environment begins in your gut. And the science behind it is nothing short of extraordinary. Here's what I'm going to cover: the specific gut bacteria that have been shown in peer-reviewed research to enhance cancer immunity and improve immunotherapy outcomes, why the modern Western diet has depleted these bacteria in most adults, the precise biological pathways through which gut bacteria communicate with your immune system's cancer-fighting cells, the exact foods with quantities that clinical studies suggest can help replenish these bacteria in a matter of weeks, and the most common everyday mistakes that silently destroy these beneficial microbes. Quick favor before we dive in. I get messages from people who've watched multiple videos and didn't realize they weren't subscribed. Could you take 2 seconds and check? It's free, and it helps this information reach more people who are looking for evidence-based health content. Now, let's start with a fundamental truth about your body that will reshape how you think about cancer. Every single day, your body generates cells with genetic mutations. Some of those cells carry the potential to become cancerous. This is not fear-mongering, it's basic biology. The real question is not whether these abnormal cells appear. They appear in every single person. The question is whether your immune system catches and destroys them before they can establish themselves. Picture your immune system's natural killer cells and cytotoxic T cells as an elite security team patrolling every hallway of a massive building around the clock. When they're well-equipped, well-rested, and well-nourished, they catch intruders before any damage is done. But when they're understaffed and under-resourced, threats slip through, they settle in, and they start to grow. Here is what most people do not realize. The bacteria in your gut are essentially running the staffing agency for that security team. They are directly involved in recruiting, training, and deploying those immune cells. When the right bacteria are present in healthy numbers, your immune system is sharp, vigilant, and highly effective at clearing abnormal cells. When those bacteria are depleted, wiped out by courses of antibiotics, diets heavy in processed food and sugar, and chronic stress, the security system goes dark. And that is when cancer gets a foothold. Now here's where it gets truly fascinating. Researchers at Gustave Roussy, one of the leading cancer centers in Europe, conducted a landmark study published in the journal Science in 2018. They examined patients with advanced lung and kidney cancers who were all receiving the same class of immunotherapy drugs. PD-1 checkpoint inhibitors. Same drugs, same treatment protocols. Yet the outcomes were dramatically different. Some patients responded powerfully, others showed no benefit at all. When the scientists analyzed what separated responders from non-responders, they found one striking biological distinction. The patients who responded well had significantly higher levels of a gut bacterium called Akkermansia muciniphila. The patients who didn't respond had very little of it. This finding sent shockwaves through oncology because it implied that a cutting-edge immunotherapy regimen could be rendered far less effective simply because of the microbial composition of a patient's gut. And they went further. When researchers transplanted gut bacteria from patients who responded to immunotherapy into germ-free mice and then gave those mice the same treatment, the mice responded. When they transplanted bacteria from non-responders, the mice did not respond. It wasn't the cancer biology alone driving the difference, it was the microbiome. Let me now walk you through 3 biological pathways that explain how this works, each mapped with real scientific precision. Pathway number 1. The gut immune axis. Your gut lining houses more immune cells than any other organ in your body. Approximately 70 to 80% of your entire immune system resides there. Akkermansia muciniphila lives in the mucus layer of your intestinal wall and physically interacts with those immune cells. It stimulates the production of natural killer cells and cytotoxic T cells, the 2 primary cell types responsible for identifying and destroying cancerous cells. When Akkermansia levels are robust, these immune cells don't just stay in the gut, they circulate throughout your bloodstream, patrolling your entire body for threats. When Akkermansia is depleted, that systemic immune surveillance fades. Your body goes from high alert to barely functioning. Pathway number 2, the short-chain fatty acid connection. Your gut bacteria don't just sit idle. They actively consume the dietary fiber you eat and ferment it into compounds called short-chain fatty acids, primarily butyrate, propionate, and acetate. These are not waste products. They are powerful signaling molecules. Butyrate, in particular, has been shown in laboratory research to suppress the growth of cancer cells by triggering a process called apoptosis, essentially programming damaged or abnormal cells to self-destruct before they can replicate. Think of butyrate as a molecular command that tells defective cells to shut themselves down. Cancer cells actively try to resist this signal, but research suggests that at sufficient concentrations, butyrate can override that resistance. The only way to produce adequate butyrate is to consistently feed the bacteria that manufacture it, and that requires a diet rich in fiber. Most adults in developed countries consume less than half the daily fiber recommended for optimal gut health. Pathway number 3. Gut bacteria as immune checkpoint regulators. This is where the science becomes truly remarkable. Your immune system has built-in safety brakes, molecular signals called immune checkpoints, that prevent it from attacking your own healthy tissue. Cancer cells are extraordinarily adept at hijacking these brakes. They display specific proteins, like PD-L1 and CTLA4, that essentially wave a white flag at approaching immune cells, tricking them into standing down. Immunotherapy drugs called checkpoint inhibitors work by blocking those deceptive signals. But here is the critical discovery. Gut bacteria regulate how sensitively your immune cells respond to those drugs. The right species, Akkermansia muciniphila, Bifidobacterium longum, Faecalibacterium prausnitzii, essentially recalibrate your immune system so it can recognize the signal that says, "This is a cancer cell. Destroy it." Without those bacteria, even the drugs specifically designed to unlock your immune system may struggle to get the door open. Now, let me share additional research that shows just how responsive these bacterial populations are to what you eat. The study I mentioned by Rauti and colleagues published in Science demonstrated through fecal transplant experiments in mice that the microbiome, not the cancer type genes, was a key variable in immunotherapy response. This was a pivotal moment in cancer research. In a separate study published in Science in 2021, researchers from the National Cancer Institute and MD Anderson Cancer Center tracked 128 melanoma patients receiving immune checkpoint blocker therapy and measured their dietary fiber intake. Among those patients, every 5-gram increase in daily dietary fiber was associated with a 30% lower risk of their disease progressing. Patients consuming at least 20 grams of fiber per day survived significantly longer without disease progression compared to those consuming less. The researchers concluded that dietary fiber was functioning as a microbiome optimization tool that helped immunotherapy work more effectively. Research into green tea has also shown promising connections. Multiple epidemiological studies, including large-scale analyses, have linked regular green tea consumption to a reduced risk of colorectal cancer. The proposed mechanism involves EGCG, green tea's primary catechin, which appears to selectively support beneficial species like lactobacillus and bifidobacterium while suppressing harmful bacteria that crowd out these protective strains. And in research from UCLA, scientists found that pomegranate extract E, which contains compounds called L-argitannins, was associated with increased Akkermansia muciniphila levels in the gut. The L-argitannins are converted by gut bacteria into a molecule called urolithin A, which appears to stimulate Akkermansia proliferation. While more research is needed to confirm exact magnitudes, the direction of the evidence is consistent and promising. These aren't vague or subjective improvements. These are measurable biological changes documented through metagenomic sequencing and immune cell analysis. Now, here is your practical protocol: what to eat, how much, and how to prepare it to support the bacteria most strongly linked to cancer immunity. Number 1. Pomegranate juice or pomegranate seeds, daily. Aim for 4 to 8 ounces of pure, unsweetened pomegranate juice each day, or eat about half a fresh pomegranate. Pomegranate contains L-argitannins, compounds your gut bacteria convert into urolithin A, a molecule linked to Akkermansia growth. This is one of the most studied dietary approaches to supporting Akkermansia levels. If the juice is too tart, dilute 4 ounces with 4 ounces of water. Consider drinking it in the evening, as gut bacteria tend to be metabolically active overnight, giving those compounds more time to ferment. Number 2. 25 to 38 grams of dietary fiber daily from whole food sources. This is foundational. Gut bacteria literally cannot survive without fiber. They starve without it. Black beans provide about 15 grams per cooked cup. Lentils offer roughly 16 grams per cup. Chickpeas come in at about 12 grams per cup. Aim for at least 2 servings of legumes every day. And here's a tip. Cooked and cooled legumes contain a form of resistant starch that specifically nourishes Faecalibacterium prausnitzii and Ruminococcaceae, the bacterial families most strongly associated with positive immunotherapy outcomes in human studies. Number 3. 3 cups of green tea daily. Brew at approximately 175 degrees Fahrenheit, never boiling, because excessive heat damages the catechins that feed beneficial bacteria. Steep for 3 to 5 minutes. Add a squeeze of fresh lemon, which research suggests can increase EGCG absorption substantially. Avoid adding dairy milk. Proteins in cow's milk can bind to catechins and significantly reduce their bioavailability. Plant-based milk is fine. 2 cups in the morning and 1 in the mid-afternoon can help maintain steady catechin levels throughout the day. Number 4. 1 cup of mixed berries daily, fresh or frozen. Frozen berries are flash frozen at peak ripeness and retain their anthocyanin content just as well as fresh. Anthocyanins act as prebiotics, selectively feeding bifidobacterium and lactobacillus species. Research has linked regular berry consumption to increased natural killer cell activity and immune activation, not from the berries themselves directly, but from what they nourish in the gut, which in turn activates systemic immune responses. Number 5. Broccoli sprouts, about 1/4 cup, 3 to 4 times per week. Broccoli sprouts contain dramatically higher concentrations of sulforaphane compared to mature broccoli. Some research suggests up to 100-fold more. Sulforaphane activates the NRF2 pathway, which helps reduce the oxidative stress that can deplete beneficial gut bacteria. It has also been shown to inhibit the growth of H. pylori and other pathogenic bacteria that compete with protective strains like Akkermansia. You can sprout them at home with a simple sprouting jar. Seed to sprout in about 3 to 4 days. Add them to salads, sandwiches, or smoothies. Avoid cooking them, as heat destroys the enzyme myrosinase that is needed to activate sulforaphane. Number 6. Fermented foods, twice daily. Have 1 serving of plain, unsweetened yogurt with live active cultures at breakfast, and 1 serving of kimchi, sauerkraut, or kefir with dinner. Fermented foods introduce live lactobacillus strains directly into your gut. The Stanford University study I mentioned earlier found that a diet high in fermented foods increased microbial diversity by 19% in just 10 weeks and significantly reduced multiple markers of inflammation, the very inflammatory environment that can allow cancer to evade immune detection.